Should we start? So welcome to the One World Probability Seminar. We are very happy to have in today's session, well, we will start with the talk of Amandine Veber. Amandine Veber is one of the central researchers in probability applied to biology. So we expect a very interesting talk, and we are very grateful that you agreed to be here today. And she will talk about modeling the evolution of genetic diversity in a population living in a spatial continuum. Thank you, Amandine.
Well, thank to you. You're way too kind with me, so no stress. So first of all, I should say thank you very much for the invitation. I'm really pleased to be here, and it's an honor, of course. And also, I should say that I won't see you because I will hide you. Like now, I still see you a bit, but then I will hide you to see my slides. And so please just open your micro and ask a question if you have a question. Okay, so my talk is meant to be a sort of review of early work on the model that I've been developing with a lot of friends now in the last decade. And so I will give a sort of overview of the questions that we wish to address in spatial population genetics, and then of the model that I've been mainly using to address them. And then Apolline will show you a more precise example of what we can do with such models. So once again, don't hesitate to interrupt me if something is not clear, and I'm happy to take questions, including during the talk. Okay, so globally, the aim is to find a tractable way to model the way genes are passed on from one generation to the next in a population that has a spatial structure, to understand how the genetic diversity evolves in such populations. So many models exist, and they've been existing for a long time for spatially structured populations. The main example is the stepping stone model introduced by Kimura and Weiss in the 60s. But most of them assume that the population is split over a discrete space. So it's split into discrete communities. In each community, the population is well mixed. There's no spatial structure anymore. And these communities or demes are related through migration. And if you think of a forest, or if you think of a field of plants, just like the one on the picture here, it's not always that easy to put a grid on such population to decide what is a community and which pair of plants belong to the same community and which belong to next door communities, let's say. And so at least for statistical purposes, or to be able to compare the theoretical predictions of the model with real data, it is sometimes much easier to start with directly in the continuum, directly with the population which is spread over a continuous geographical structure. And it is what we're going to be interested in this talk. And so from now on, I will have a continuous spatial structure. Biologically speaking, the most interesting dimension is 2, of course, like the populations are spread over a 2-dimensional space, but mathematically speaking, the model will be interesting in any dimension. And so most of the time, we really look at any dimension and try to derive mathematical properties of the model in dimension D, let's say. So what should be the footprint of a spatial structure? What do we want to put in those models? Then first, interactions or reproduction should be between sufficiently close individuals. If I come back to my picture here, each flower is going to have a certain number of offspring, but most likely these offspring will happen around in some more or less extended neighborhood of their parents. And it's very unlikely that this flower that you see here will send an offspring directly on the other side of the range of the population. Okay, and so interactions, competition for resources, and all these evolutionary forces should happen more or less locally. And this is what we want to include in model with a spatial structure. And offspring should be born in a more or less extended neighborhood of their parents. Of course, it can be a large neighborhood of the parents if you think of plants that send their pollen or seeds in a long way away due to wind or pollinators, for instance. But what you want to encode is that in some way, the offspring are born around their parents. Yeah? And what you can incorporate as well in these models is the fact that the environment may not be the same everywhere in space. If you think of a mountain range, for instance, the conditions in a valley are not the same as the conditions in higher altitudes. And so the selective advantage of some genes compared to, or some alleles compared to others, may differ depending on where you are in space. So I will use the word gene. So we'll be focusing on one region of the genome, say a gene that codes for something, and the different versions of the genes will be called alleles, and I will use types as well from time to time. And so we'll really focus on at one gene, say, which can have several versions, and we want to understand the genetic diversity of the population, so the variability of the alleles that you observe at this gene throughout the population. And so the results of this spatial structure should be that if you look at the local allelic distributions, they should be correlated in a specific way that depends on how individuals reproduce, how far away they send their offsprings, etc. And so the correlations between these local allele distributions will depend on parameters such that the speed of spatial diffusion of a gene across the population, so the speed at which genes are passed on from one parent from parents to offspring, sorry, and offspring are not exactly at the same location in space, and that makes the genes sort of move in space. Okay, and the questions of interest are like statistical ones, like can I identify compound parameters that characterize the genetic diversity in a population and how it evolves through time? Can I characterize the correlation patterns between the local allelic distributions at several locations, depending on the evolutionary scenarios that I consider, whether there is natural selection, whether everyone has the same chance to reproduce or not? And of course, can I reconstruct from particular data the key parameters that I have found in my first point here? And also from a statistical perspective, like if I give you a sample of genetic diversity, like the one you see on the, if I give you this pattern here that you see on the picture, can you tell me whether the evolution is neutral in the sense that everyone has the same chance to reproduce, or whether some evolutionary forces are in action? And of course, it is not usually is not as easy as on this picture. So to answer this question, I will use the spatial lambda Fleming-Veaux process. And so this process was introduced by Alison Etheridge and Nick Barton. Alison is a mathematician in Oxford. I guess most of you know her. And Nick Barton is a biologist at IST Austria. And they introduced it in 2008, sorry. And it was the sort of main goal of my of half of my PhD, at least, to develop like first examples of how to use this model, pose this model a bit more rigorously, and show first examples of how to use it to study the genetic diversity in a population. And after that, a lot of colleagues kept working on it, and so I will not cite everyone, but there are obviously contributions by Apolline Louvet, but also by Sarah Pennington, Nick Freeman, and Nathaniel Berestiki, and I should say it's Jérôme Kelleher as well. So a lot of people. So in my population, so think of my population as being a density 1 of individual everywhere in space, and each individual will have an allele or a type, as I told you. So my allele space will be K in general, a compact set. Most of the time, I will take K to be just the set with 2 possible alleles, 0 or 1. Just for simplicity, but it's a caricature of what happens for any K, actually. A lot of behaviors can be understood only with K with only 2 elements, in fact. And then at time T, my population, the state of my population, will be represented by a measure, MT, on space, so RD, geographical space, times allele space, K, whose first marginal is Lebesgue measure on RD. Okay, so you have a uniform density of individuals everywhere in space, density 1 of individuals everywhere in space. You think of a very dense population of plants because they won't move during their lifetime. So density 1 of individuals everywhere in space, and at every location in space. So Density 1 is encoded by this Lebesgue measure here. And at every location X in space, you have a distribution of alleles at site X at time t, which is rho t of x dk. Okay? So the measure in which I'm interested is this measure mt on space times alleles, but I can always decompose it as Lebesgue measure times a kernel rho t, which tells me if I sample someone at site X at time t, what's the distribution of its allele, its type. Okay? And so this is how I represent my population at time t, but now I need to make it evolve as time goes on. And in this model, the characteristic is that usually what you do is that you attach a little clock, an exponential clock to every individual in your population. And whenever it's an individual clock rings, then the individual reproduces and kills someone else, etc. Here, to sort of impose some local regulation of population size, we took another point of view, which is to say we're going to have a Poisson point process of reproduction events that are going to fall over space and time in some way. Okay? And these events, when you specify where and when they take place, after that, you have to specify what exactly happens during your reproduction events. But instead of having individual clocks where individuals drive the reproduction is going to be based on a sequence, a random sequence of events that specifies where and when something happens. And then you have to say what the something is, what happens during your reproduction events. So for that, we need a measure mu, which is sigma finite on R plus, like we exclude 0. So it's going to give us an intensity on radii for the events. You also need a collection of probability measures on 0, 1, which I call nu R. They are indexed by the radius of the event that will be sampled by mu. And we let pi be a Poisson point process on R. So this is time times RD. It's going to be the center of the event times R plus the radius of the event times 0, 1. It's going to be the impact of the event. It's going to be the fraction of individual replaced during the event. And this Poisson point process has an intensity measure, Lebesgue measure on time and space times mu of dr, mu r of du. So events fall uniformly in space and time, but then you can play with the radius and you can make the impact, so the fraction of individual replaced during the event, you can make it depend on the radius of the event itself, with the idea that for a small radii, it's going to be like they are going to encode very local reproduction with a small impact. So you expect a small u to go with small events, say spatially small events, whereas large events, events with a large radius, are sort of catastrophic events in which you're going to replace a larger fraction of your population. But of course, you can play with the two and encode the situations that you want. And so you have a random sequence of quadruplets, T, X, R, U. Okay, so this is pi. And if I take one of these points in pi, then at time t, in the ball centered at x with radius r, what you do in the simplest version of the model is that you choose a parental allele kappa according to the allele distribution just before the events within the ball B of xr. Okay, so everything happens in this ball. It's going to be the place, the location of the reproduction events. Nothing happens outside this ball. And you take a parent, in a sense, you take a parental allele kappa according to the type distribution, allele distribution in the area of the event just before the event. And then at every location in the ball B of XR, the new distribution of alleles is made of as follows. So you keep a fraction 1 minus u of the population as it was just before the event. And then you replace the fraction u that you've withdrawn with offspring of the parent chosen. So you replace the fraction u of the population by individuals that are all of type kappa. Okay, so you have this random sequence of events, and then during each event, in the area of the event, you pick a type, and then you replace a fraction u of the individuals there by offspring of this type. So individuals which are all of a type kappa. So it's not obvious that such a process exists, because if you think of it like in any finite time interval, there's an infinite number of events occurring, because I have Lebesgue measure. So the number of events that happen somewhere in this infinite space RD is infinite in any finite time interval. But we first proved the existence and uniqueness of such a process using a method developed by Steve Evans on continuum sites, continuous space stepping stone models, which is more analytical in the sense that you construct the semi-group by a duality property that I will speak about later. But you can also have a more constructive way of saying, okay, I wanted to represent a population of individuals going through my reproduction events. And so you start from a Poisson point process of individuals, like discrete individuals, and they go through the sequence of reproduction events that I have told you about in the previous slide. And if you make the density of these individuals go to infinity, their empirical distribution converges to the process that I've told you about. And this is something that we proved with Anton Backolbinger in a paper in 2015. And a more general construction was proposed by Etheridge and Kurtz, and it was in 2019, but actually the ideas evolved at the same time. It's just that it took them a bit longer to publish it. And so a sufficient condition, not necessary actually, but sufficient condition for the process to be well defined is this integral here to be finite. So I'll come back to it later because it has an interpretation, but what you want is that the integral over R plus and the integral over 0, 1 of u r to the d, nu r of the u, mu of the r to be finite. And in this, under this condition, there is a process with values in the space of measures on RD times K, whose first marginal is Lebesgue measure, and that does what I showed you earlier. Okay, so this state space here, we endow it with the topology of Veber convergence, which means that when I will integrate my measure, I will always integrate it with respect to or against a function with compact support. The reason is that there's a density 1 of individual everywhere in space. And so the integral of 1 is infinite, and the integral of any function that's going to have like non-compact support has the risk to be infinite. And so we really have to work with neighborhoods or compact neighborhoods rather than the whole space. We cannot use the weak convergence in this framework. And actually, I give you one version, the sort of easiest version of the model, but you can replace, what's key in this model is the Poisson point process of events. The reproduction encoded by a Poisson point process of events. If you don't like balls, you can have squares, you can have any shape you want, or you could have Gaussian replacements in the sense that you choose a center for the reproduction, and instead of choosing a radius, you choose a variance, sigma squared, and you choose a parent around the center of the event according to a Gaussian kernel with variance sigma squared, and then you replace individuals according to the same Gaussian kernel or another one, etc. And you can also add natural selection recombination. You can have inhomogeneous environments, so you can play with the model and add little bricks to it the way you want. What's really key in this model is the phrasing of the reproduction events as a random sequence of events driven by a Poisson point process. So pictures now to make it a bit more concrete. So these simulations were done by Habib Saadi, who was a PhD student with Alison at the time. And so the radius is going to be fixed to some small value. I don't know which one. And you replace u is 1 all the time, which means that in every reproduction event, you replace every 1 all the time. Okay, so the fraction of individuals replaced during a reproduction event is always 1. So you start from a configuration where everyone is of type 1 here, say, and everyone is of allele or type 0 here. And then after 2 times 10 to the 6 events, this is how the interface looks like. And you can keep going. And you see that the interface become wider. You have islands of red in the ocean of blue, and conversely, islands of blue in the red region. And so the interface becomes rougher and rougher. It becomes wider. And of course, I have only two colors here because I replace everyone during each event. But if you were 0.5, for instance, I would also have a gradient of purple across the interface here. Okay, so this is typically the kind of situation that we want to understand. What's the long-term behavior of such interfaces? How fast does the interface grow? How rough does it become through time? Or can we get a more like first-order picture of what happens, etc.? So before I go in this direction, I will introduce a very convenient tool that everyone knows in population genetics because it is very standard. When you want to understand a population model, where a model where individuals reproduce and transmit their types or alleles to their offspring, a key approach is to think about genealogies. And usually it's useful because then you can chase duality relations between the forwards process of reproduction, the forwards meaning that you go from the past towards the future, and the dual process, your potential dual process at least, of genealogies, in which case the time goes from present towards the past. So the arrow of time is in the other direction. So let's think about what it is to follow an ancestral line in our model. So suppose you take one individual in your population at time t. Your process has been evolving for how much time as you want. And you pick one individual, say at location 0, and you want to come back into the past and you wonder where its parent was. Okay? And if you do that, then you have to wait for some time. You come back into the past slowly, and then suddenly your individual is in the area of a reproduction event. And maybe with probability U, it was in the fraction replaced during the event, which means that your individual that you picked at time t was actually an offspring of the one chosen to reproduce at the time. Okay? And so if you make this mental experiment, then you come back into the past, you find the first reproduction events in the past in which your individual is in the area covered by the reproduction event and is in the fraction of the population replaced by offspring of the parent. This parent was chosen according to the allele distribution in the ball. So its position is essentially uniformly distributed over the ball. And so if you follow the location, the spatial location of the ancestor, of your ancestral line, then you wait for some time at 0, say where you sampled your individual, and then suddenly in the past, you jump onto the location of the parent, which is not exactly the same as that of the offspring. And then you keep going back into the past. You're going to be at some point in another reproduction event during which your individual was, or your ancestor was among the fraction replaced. And so it has another, like a different ancestor, a parent, which was chosen uniformly over the ball of the event. And so there's another jump, etc. And so if you keep going into the past, coming into the past, then what you see for a single ancestral line is a jump process, which is symmetric because your parents, everything is sort of translation invariant in my model. So there's no reason why it should jump more to the left than to the right, essentially. And so you have a symmetric jump process. And now if you think about several, so sampling more than one individual at time t, then what happens is that unless they are hit by the same reproduction events in the past, then they move in space a bit, not independently, but at least they don't use the same reproduction events to move. And then suddenly they're going to be maybe in the area of an event, and both in the fraction replaced during the event, in which case both of them come from this parent, the single parent chosen during the reproduction events. And so their ancestral lines merge into this common ancestor, and you keep going back in the past to find common ancestors to the whole sample. Okay, so if you sample like 10 individuals in your current population at time t, and you trace back where their ancestors come from, and where in space their ancestral lines are, as you go back into the past, then what you obtain is a system of correlated symmetric jump processes which coalesce or which merge when they are affected by the same event. Affected being in the same, all in the same fraction of individuals replaced during a single reproduction event. Okay. And so what's the rate at which an ancestral lineage jumps when it's presently at 0? Say it's all translation invariant, so 0 means nothing here. But it's the rate at which your individual is in the area of an event and is in the fraction you replaced during this event, integrated with respect to the intensity measure here. And so if you integrate first over the x coordinate, this integral here becomes just the volume of the ball of radius r in Rd. And so you have the integral of this double integral here of u here times the volume of the ball of radius r, nu r of du, mu of dr. And if you remember, I asked that this should be finite, and it was a sufficient condition for the model to be well defined. And this is where it comes from. Under this condition here, if this quantity here is finite, and a single ancestral lineage is jumps at finite rate. So it's just a compound Poisson process, okay, symmetric compound Poisson process. And then the miracle comes. So I give you the duality formula only in a simple case where K is made of two types. A more general formula exists, but it's much more notationally heavy. So and it says exactly the same thing as here. So suppose that K, there are only two alleles, 0 and 1. And WT of X is the frequency of allele 1 at site X at time t. Okay, so 1 minus WT of X is the frequency of type 0 individuals there. Then if for any J, for any integer J, and for any psi, which is continuous and compactly supported over Rd to the J, so any kernel to pick J locations on Rd, say, then the expectation for the forwards process, the spatial lambda Fleming-Vio process of psi of x1, x2, etc., xj times the products of wt of xi, dx1, dxj. So this integral here, which is where the expectation is with respect to the forwards process, measure valued process, this is equal to the integral of psi of x1 up to xj of the expectation for the dual genealogical process, starting from one individual, one lineage at x1, one lineage at x2, etc., and last lineage at xj, of the product over the number of ancestors t units of time in the past, of w0, so w at time 0, of at the location where the ancestors are. Okay, so it looks a bit complicated, but when you think about it, so when you forget about the psi's here, so the psi's are just here to make the formula rigorous, but it is valid for any psi. So for almost every x1, x2, etc., xj, if you forget about the psi, the integral with respect to psi here, what it says is that the expectation for the forwards process of the product, which is here, so the probability that if I pick someone at site x1, someone at site x2, etc., at time t, they are all of type 1. It's the same as the probability if I start my ancestral lineage, my ancestral process from one lineage initially at x1, one initially at x2, etc. I trace back t units of time in the past. So I come back to time 0. And I check that all the ancestors, where they are in the fraction of type 1 individuals where they are in space. Okay, so this really says only that my individuals today at time t are all of type 1, if and only if all their ancestors, t units of time in the past, are all of type 1 as well. Yeah, this is all this formula says. And it's very convenient because And WT is rather, it's a field of random variable. It's not very easy to attack directly. And whatever you can learn on these systems of correlated symmetric jump processes, you can transfer this information onto the forwards process and transform information on the dual process into information on the forwards process. And that's where the point of view that we will use later. So if you remember my like the simulations I showed, which are not mine, they are Habib's, we were starting from everyone of type 1, say, like blue to the left, and everyone of type 0 or red to the right. And we were looking at the evolution of the interface as time goes on. So let's look at what we can say about this situation. So this is joint work with Nathaniel Berestiki and Alison Edridge some time ago now. But so our geographical space is still R to the D, not only dimension 2, RD. Okay? Only two alleles, 0 and 1. And I will look at two cases. The first one is constant radii. So we fix a radius r, we fix a fraction, an impact u. And so all my events will have a radius r and impact u. Okay, so you remember they are still random because they can fall anywhere in space and time, essentially, according to Lebesgue Poisson point process with the Lebesgue measure, but they will all have a radius r and an impact, a fraction replaced u. So in this case, reproductions are purely local. And something that was Nick Barton was interested in was to have catastrophic events, rare, but with the macroscopic consequences, say on the dynamics of the population. And so we considered the case of a heavy-tailed radii. So we fix alpha, a parameter alpha between 1 and 2. We fix u, so the impact will be fixed, but the radii will change from one event to the other. And the intensity measure on radii is going to be given by mu of dr, which is essentially dr over r to the d plus 1 plus alpha plus 1. And the indicator function that r should be bigger than 1 here is just for technical reasons, because you cannot accumulate two small jumps. Otherwise, it violates the condition that I showed you. So this allows the occurrence of rare but very large events. For instance, forest fires that happens every like hundreds of years, or glaciations that happens every 10 to 10,000 years also. And the reason for this, we could have encoded this in many ways, but we chose this one because in this case, ancestral lineages behave like alpha stable processes, and they are therefore more like a bit easier to manipulate. And of course, we're going to look at very long time scales. So time will be scaled by a factor n. So we'll accelerate the movie by n, say. And a game that we play a lot in population genetics is to find the right space and time scales. And so I tell you, and you have to believe me, that the right spatial scale to consider is n to the 1 over alpha. And in case 1, when the radii were fixed, then I set alpha to be 2. And there you recognize that in the fixed radius case, you accelerate time by a factor n, and you scale space by square root of n, and it's going to look very much like the Brownian scaling. Okay, that's nothing surprising here. So you accelerate time by a factor n, you look over very long time scales, but because there are long time scales, you also need to look from a satellite, look from very high up, okay? And so you scale down space by a factor n to the 1 over alpha, and this is this distribution here, this random field of probabilities of being type 1 in space that we're going to consider in what happens, in what follows, sorry. So we start from the interface that I showed you earlier. Everyone is of type 1 to the left of x1 equals 0, and everyone is of type 0 in the other half plane. And the question is, how does WTN behave when n becomes large? What's the width of the interface? What's the resulting pattern of genetic diversity, etc.? So the answer is the following. If you look at the first case, where all events have radius capital R, And here U is set to be 80%. So in each event, you replace 80% of your local population. The radius is given by this quantity here, and the scaling parameter n is 1,000 here. And these simulations were done by Jérôme Kelleher, who's still in Oxford. You start from this step pattern here. What you see after 10 to the 5 events is that you still have a step pattern, but it moved a bit towards the left. And after 10 to the 7 events, you still have a step pattern, but it moved this time to the right. Okay? And so what you can sort of anticipate is that the answer here is that you will keep this step all the time, but the interface, the location of the interface will move like Brownian motion, because there's no reason why it should go more to the left than to the right. So yeah, you're going to keep an interface here, a sharp interface, and the location of the interface is just going to move like according to Brownian motion. And the way you formulate these results is there exists, so as n tends to infinity, as my scaling parameter tends to infinity, there exists a process, mt2, with values in m lambda, such that the finite dimensional distributions of mn converges to m2. And you cannot have tightness, actually, because you have a lot over these timescales, you have a lot of teeny tiny events that have a macroscopic, that drives a macroscopic jump, because you replace 80% of your population, okay? So you cannot have tightness, but you can have convergence of the finite dimensional distributions of the process Mn towards the process M2, which can be described as follows. So there exists a parameter sigma squared, sigma tilde squared, such that if x is a standard Brownian motion, and if I define pt2 of x as being the probability starting from location x that my Brownian motion at time u times sigma squared t belongs to the original area of ones. So pt2 is the probability that Brownian motion run at the right speed comes back to the area of ones in t units of time. Okay? Then in one dimension, WT2 is a random field of correlated Bernoulli random variable. So everywhere in space, either everyone is of type 1 or everyone is of type 0, and there is no other option. Right? And the probability that everyone is of type 1, of allele 1, is given by P2T of x. And in higher dimension, the limit is deterministic and is given by, is equal to P2T of x. So this is just a heat kernel. When there are catastrophes, again, these are simulations by Jérôme Kelleher, you replace 80% of your population in each event. Alpha is taken to be 1.3, and this time n, the scaling parameter, is 10,000. You start from this initial condition here, which mimics what happens when you have a first big event, say, and then you look at how the very rapid small events like deal with this new step created by a big event, say. And so what happens after 100 events, you can see it here. And if you zoom on the interface, you start to see that somehow we're going to go back to a 0-1 picture. And after 10 to the 6 event, this is the genetic diversity in the population and the pattern of allele frequencies. And if you zoom on the interface here, what you really see is that either at every location in space, either everyone is of type 1 or everyone is of type 0, and there is no intermediate, okay? And so, and the same happens this time in higher dimensions. So in 2 dimensions here, if you have a major, like a big catastrophe, let's say, and if you let the small events happen very quickly, what happens is that they quickly restore the 0-1 pattern, and everywhere in space, you see only black or white, meaning that there is only type 1 or only type 0 individuals everywhere at every location in space. Okay? And so you can formulate another theorem, just like the previous one, except that now the finite dimensional distribution of Mn converge to another process, the finite dimensional distributions of another process, which is described in terms of a symmetric alpha stable process X alpha. And if you call Pt alpha of X the probability that starting from location X in space, your alpha stable process run at speed u comes back to the initial area of ones, then in any dimension, WT, the limit here, is made of a random field of correlated Bernoulli random variables that satisfy that the probability that everyone is of allele 1 at site x at time t is given by pt alpha of x. Okay, and so I won't show you the proof, but really what you should see here is that this is tracing back an ancestral line, and the probability that everyone at site x at time t is 1 is the probability that if you come back into the past, actually everyone, the core of the proof is to show that if you take several individuals very close together, they coalesce, they find common ancestors very quickly, and then the probability that this ancestral line traces back to the area of where you have type 1 individuals gives you the probability that locally you observe type 1 individuals at time t. Okay, and just to finish, so the conclusion to be quick, there's no local coexistence of alleles except in more than in dimension greater than or equal to 2, and reproductions are purely local. Okay, so these were the only cases where you could have something in between 0 and 1, and which is not 0 or 1. Okay? And also this analysis tells you that local allele frequencies are correlated through the transmission of alleles, or the process of transmission of alleles, and the correlation length is square root of n in the case where reproduction is only local, and n to the 1 over alpha in the case where you have this heavy-tailed radius distribution. And so rare but massive extinction recognition events can really have significant impact on the genetic diversity seen in the population, even though they are rare. If they are macroscopic enough, in some sense, they do leave a trace. So just to finish with a sort of an example that I wanted to show you, and then we'll be done. So this is a topic that I've been discussing with Marianne Felix and Enrique Teotonio from École Normale Supérieure, from the biology lab of École Normale Supérieure. So we discussed this some years ago now, and we couldn't really go much farther than quite a qualitative discussion, let's say, but at least it was interesting to see this example. So this little guy here is a C. elegans. So it's a little worm. It's 1 millimeter long, and its life, so it's a model organism in biology. So we know absolutely everything about its life in laboratory, but it's much less known what happens in nature. And this is one of the topics of Marianne, to understand the genetic diversity of the wild populations of C. elegans. So this nematode has a complex life cycle. I won't give you all the details, but what happens when they don't have enough food is that instead of growing into adults, they can grow into a dour state. So dour is a sort of dormant state where they cannot eat or reproduce, but they can move. Okay, so they are not actually dormant. They move in space, but they don't do anything else. And their population dynamics happens mostly in fall, and they grow on rotting plants, say. That's because they feed on bacteria that are in the rotting plants. And what happens is that when a food source appears, when the plant starts rotting, the few nematodes that manage to reach this source of food develop exponentially fast. So you have an exponential growth of population based on a few individuals that are there, that are around. And when the resource becomes exhausted, the new eggs, say, instead of becoming adults, they turn into this dour stage, and then they move around to find another source of food. Okay, and migration is possible via individual motion, which is slow because they are 1 millimeter long, and they live in a difficult environment where there's mud and all sorts of plants, etc. So it's not so easy for them. But they can also hitchhike in groups. So they hang on a snail or an isopod by like packets of 10 individuals, say, and these bigger animals can bring them several meters away. And actually, they seem to be able to sense when it is the right moment to jump out of the snail because there is food around. Okay. And these are samplings done by Marianne in Santeuil, so it's in the Paris region, and she did that once or twice a year for something like 10 years. And then she genotyped some of the nematodes that she found. So what you see here is a river, okay, and then there's a train line here. railway, right? And you see different haplotypes of nematodes. A haplotype is, sorry for all the biologists in the audience for my caricature, but essentially a haplotype is a group of individuals that share exactly the same DNA at the sites in which you look. So it's not whole sequencing. You just look at particular sites on the genome, and the haplotype is a group of individuals that has exactly the same DNA at these sites. Yeah. So there are orange, green, and blue, and the red is another species called Cenorhabditis brixae. And what you see is that locally, you see a lot of green here, a lot of orange here. You sort of see some clustering of genotypes or haplotypes, at least, along this river here. So what I'm I'm a bit, cheating here, because I'm sort of suggesting that it is the one-dimensional clustering that we observe in the results with Nathanael and Alison. Of course, if you look at the scale here, this is a 10-meter band of land, and for a nematode, which is 1 millimeter long, it's not particularly one-dimensional. Okay, so we are not really in one dimension. But if you think about the proof, actually, the proof says that if you look at the genealogies in one dimension, they cannot cross without going close to each other. And because these worms reproduce by boom and bust like this, it is not unlikely that actually, if you trace back the ancestry of a sample of worms, then they really have to go through the same rotting plant to then pass each other, like, geographically. Okay, so it's not like the worst limitation of my example, let's say. And anyway, you cannot really, I mean, we don't know the evolutionary forces there. Probably there's a lot of competition for food and resources, and there's a lot of like difficulties in surviving, etc. So most likely there is already some spatial segregation, which is due to competition, and the boom and bust dynamics just reinforces this selective pattern. And so that's why we did not go further than this. And also a last thing that I should say is that she did 1 or 2 sampling each year, which is complicated enough. I mean, all the merit goes to her. And Marianne Felix. But of course, I showed you the most obvious pattern. You know, there are some other samplings in which it is less of a leap of faith to talk about spatial segregation. You see a lot of blue here or green, but anyway, so but these boom and bust dynamics are thought to create spatial segregation, and so I thought it was a nice example to show you to connect the theoretical model with some real life biology, let's say. And you can add natural selection in this model, and Apolline will tell you how. So I kept the slides, although I thought I would not have no time to present them. So if you want to come back to it during the questions, we have results on natural selection, and we have like results with Alison and Feng Yu, in which we recover the Fisher KPP equation, a measure-valued version of the Fisher KPP equation based on the model with weak selection. And when you add long-range dispersal, we get the Fisher KPP equation, but with the Laplacian replaced by a fractional Laplacian. And this is the result I'm showing you here. And just to name a few perspectives, there's a lot of parameter regimes to explore, a lot of forms of selection and competition, etc. And there's already a lot of work done by Alison Etheridge, Sarah Pennington, and other people. You can also, when you add selection, you can wonder about what's the effect of selection on one gene on neutral genes that are on the same chromosome, let's say. So it is a big question in population genetics. What's the footprint of natural selection on the whole genome and not only at the gene that you consider? Of course, there's a lot of statistical questions associated to it. How do you do parameter inference? Can you reconstruct the origin of the intensity of a selective sweep, or like an invasion of an allele because it's favorable to its carriers? And Apolline is going to present a modification of the model with selection, where you choose K parents, and if all of them are of type 1, then the offspring is of type 1. And it gives us a family of models for range expansion. And there, a very interesting question is what we call gene surfing. And I encourage you to look at papers by Halacek, Nelson, etc. Gene surfing is the fact that some slightly deleterious allele can remain in the population, in an expanding population for some time, just because it is surfing on the wave of expansion in some sense. It is slightly deleterious, but because the population is expanding, coming on new ground, let's say, it has some chance to remain in the population for a long time. And these are very interesting questions, but also very difficult questions. And with that, I'll be happy to take any question. Thank you very much for your attention.
Thank you, Amandine. That was really nice talk. And now there is time for questions.
Sorry, you have told that if dimension is 2 or larger and the rule are local, then there is coexistence of in space coexistence. What does it precisely mean? What is the theorem?
So it means that over the spatial scales that we consider, we just see a diffusion of genes. Okay, so it's really the heat kernel in this. So you start from a half space of ones, a half space of zeros. In one dimension, you just have the interface that moves like this. In more than one dimension, you have a mixture. The two populations somehow meet, and if you look at the frequency of type 1 individuals, it just diffuses through the type 0 individuals, and the other way around, like the type 0 managed to diffuse slowly. So slowly the two populations mix together, right, according to the heat kernel. That's what it means. And it is the only situation where you don't see a pattern where everyone is 0 or everyone is 1.
Why local? If it is non-local, it is the same or not?
No, when it's not local, when you have these very big events, then the picture remains 0-1-ish. Okay, so if I come back to the simulation, you see here in more than so I have only two dimensions, but after some time, you only see white or black. Black means everyone is of type 1. White means everyone is of type 0. Okay, and the reason for that, I guess, is that so the way you prove it is that if you take a sample of individuals somewhere in space over the spatial range that you consider, okay, they will if you take them very quickly, in all the cases where you see a 0-1 pattern, these ancestral lineages will merge very quickly. So they will have a common ancestor very, very quickly. And so this ancestor is going to be 0 or 1, but there's no other option. Whereas in the case where you have local reproduction events, but and in 2 dimensions or more, actually, if you take a sample of individuals, like 3 individuals, then there's a chance that their ancestral lines never cross. And so they just independently of each other, they're going to come back or not to the original area of ones. Okay, so it's really the fact that in all the cases where you see a 0-1 pattern, the way you prove these kind of results is that you show that individuals that are very close in space actually find a common ancestor very quickly. And so either this ancestor is a 0 or it's a 1, but there is no in between.
Sorry, for non-local case, it is a theorem or it is only picture? What is a theorem?
It's a theorem as well, like this one.
Well, but
so in any dimension, if you look at the field of allele frequencies, so WT of X is the frequency of type 1 allele 1 individual at sat X at time t, then every WT of X is a Bernoulli random variable. And the probability that this Bernoulli random variable is 1, so everyone is of type 1, is given by PT of X. So it is a theorem. And really the proof is that over these scales, because of the large events they are rare, but they are able to catch back ancestral lineages that would have diverged in space, because they happen sufficiently often that at some point they will manage to bring together lineages that have separated by some large amount.
Sorry, and for local case, it is not Bernoulli, or what is the difference between local and non-local?
So for the local case, it is Bernoulli in one dimension, again, because the ancestral lines find common ancestor very quickly, and in more than one dimension, it is deterministic and just given by the heat kernel.
Ah.
Yeah. Okay.
Thank you. Understand. Thank you.
So I think there is a question by Anton.
Hi, Amandine. Thanks for
Hello.
this nice talk. I have a question concerning in both your theorems, you had only finite dimensional distribution, convergence in finite dimensional distribution. And you mentioned that one will not expect more. Could you maybe comment briefly on that again? Whether maybe it's a simple reason.
Yeah, well, the reason is that, so you crush space. So of course, the impact of one single event is sort of small, but you replace, say, a half of your population during an event. And so you make a sort of, how to say that? You have over the time scale NT, you have of the order in a volume 1 region, you have of the order of N. So in 1 unit of time in your new units, you have of the order of N events, and they all have a macroscopic impact on your genetic diversity. And so it's sort of jumping all over the place all the time. And that's a bit what is shown here, actually. Well, it's more obvious in this representation. You see, like, you have a lot of very rapid small events that locally brings you down to 0 or up to 1 very quickly. But somehow instantly you start from this, and instantly you end up in the limit, instantly you end up with a pattern like this. So you cannot have tightness.
Some sort of separate time scales, right?
Yes, exactly. But so if you look at the picture at one given time, positive time, you will see a 0-1 picture. But the way you create this 0-1 picture is really instantaneous on this long time scale. And so, and by accumulation of teeny tiny events, that brings you back to 0-1 very quickly. So you cannot have tightness.
Thank you.
So we have maybe time for 2 short questions by Andre and then Franz.
Two short questions. Firstly, I would ask, could anything more specific, you showed the slide on asymptotics, be told about scaling limits and phase transitions of this process? Are there any phase transitions possible? And secondly, you mentioned topological aspects. Would it be possible that, because these species live in a spatial continuum, the topology of this space could have some additional influence on the stochastic process that you have?
Okay, for phase transition, I don't think they are here, because it's somehow you would need some crucial parameter to vary, and the behavior is always the same, except that the U here can be taken to be different. Or so I don't expect a phase transition, in the sense that I don't expect for U small enough, you will see a given pattern, and for U big enough, you will see another pattern. It's continuous in the parameters in some sense. So you don't expect phase transitions there. Okay. And for the second one, well, the stochastic geometry is an interesting question. We started thinking about it and then we abandoned. But so how, I don't know. There you don't, in the neutral case, you don't really expect the geometry itself to have an influence. But if you had a bit of natural selection, or if at the interface there was a reaction one way or another, then indeed the topology, or at least the borders would matter, and you would have interesting phenomena going on here. So, but just like understanding the Hausdorff dimension of this, because you see a lot of little packets of black or white, etc. This has a fractal structure in some sense. So it would be interesting to understand this pattern.
Okay, thank you.
As to detecting selective sweep, did somebody try to use the statistics that are commonly used for discrete population models? And how do they behave compared to spatially continuous models?
So I'm not sure I can really answer this question, because we've looked at selective sweeps in another work, but we haven't done the statistical part of it. So I can't really, I'm not sure I can. Most likely when the first try is to try to use the discrete space statistics and see whether they have an alternative here in this in the continuum. But I don't know about people having done that. So I don't think this has been used for inference or detection purposes. This model in the continuum has been used for detection purposes at the moment. Well, that's all. Sorry, but of course, I mean, there is no real qualitative difference between the discrete space and the continuous space over these large spatial and temporal scales. Okay, you expect the same kind of patterns. It's just that statistically speaking, it's easier to handle data which are basically continuous space if your model is already continuous space, and so you want to. It's tempting to have a continuous space model, but you don't really expect the discrete space or continuous space to make a difference over very large spatial scales, because the rescaling in space, looking from a satellite, is going to erase a lot of the details of the discrete structure anyway. So.
You can probably use statistics like isolation by distance and compare different regions of the genome. Yeah,
but so this you can do even without selection. Actually, you can. Yeah. You can infer parameters. So we've tried to do that, where inference of the gene diffusion, the diffusion speed through space, and of neighborhood size based on identity by descent. Yeah. So you can compute the identity by descent, and then you can use that to try to reconstruct the neighborhood size and diffusion.
It's just the variation along the genome that tells you about selection.
Yes.
This kind of sets.
Yeah, exactly. And that's what I will tell you about. If you go along the genome and you want to see the impact of a gene selected by natural selection on the neighboring neutral or supposedly neutral genes, and that's going to give you methods to detect the selective sweeps.
Yeah. Thank you.
Sorry, and what is the result for selection, for the case of selection pressure? What is the result?
We should continue to the next talk, and maybe we postpone this discussion after the Apolline, because so thank you very much.
Thank you
for the great talk. That's really interesting. And now we need a couple of minutes to change the slides. Apolline, if you want to.
Yeah, I just also need to. Yeah, because like 2 UK adapters, and I mean 1 UK adapter, and I need to connect 2 things to the sector. So a bit of an issue. So the slides are here.
Great. So I think we should start right away. So our next speaker is Apolline Louvet. She's a postdoc in the University of Bath. Before, she was a PhD student in the group of Amandine Veber, and she will talk about constructing a population genetics process for population expanding in a spatial continuum. Thank you, Apolline, for being here today.
Okay, so thanks for the introduction, and thank you for the invitation. So I'm very happy to present here. So what I will present you today is based on my PhD work with Amandine, and it's about constructing population genetics processes for population expanding in a spatial continuum. So indeed, like in the talk of Amandine, we saw that spatial land-Fleming processes were like fairly new models, which are very well suited for studying genetic diversity in populations living in a spatial continuum, and fill the gap in like, the family of models in population genetics. You can use it to study like a wide variety of situations in terms of genetic diversity and how does it evolve over time. You can consider neutral genetic diversity. You can add a wide variety of weak selection mechanisms. And you can also consider, for instance, like fluctuating selection and things like that. So you have like a rich variety of results. But in all this list, there is something that you don't see, which is the case of spatially expanding population. Indeed, if you remember correctly, like the state space, which was introduced by Amandine, you have a kind of underlying assumption that your population is like uniformly spread in the whole environment, in the whole space. So you don't have empty areas. And so in particular, you can't see expansions. Studying genetic diversity in expanding populations is a very interesting question from a mathematical viewpoint and from a biological viewpoint, because you expect like the results to be very different. And this was demonstrated in particular in a series of experiments by Alacek and Nelson and collaborators, where you take like colonies of bacteria or yeast with a fluorescence, which can be, for instance, here, green or red. And this new fluorescence is completely neutral. You can just see it as a marker. It doesn't influence the behavior of the bacteria or anything. So you take like your green and red bacteria, you mix them together and put them at the center of a petri dish. And you let them grow. And what you observe is that very quickly at the front edge, but on a fairly wide front edge, kind of fairly generous definition of front edge, you can see like the spontaneous separation of sectors. So in some areas, all the bacteria will be green, and in others, all will be red. And again, that's not due to like red bacteria wanting to be together or something like that. No, it's just something which occurs spontaneously. And qualitatively, it's well understood. Like what happens is that at the front edge, you have very low population densities. So you have a lot of genetic drift. So you're going to have strong stochastic fluctuations of the frequencies of each type, and you are going to lose one type randomly due to these fluctuations. And this is then going to be enhanced by the fact that at the front edge, you have like almost no competition because there is almost nobody, and you have a lot of resources available. So you have very high reproduction rates. So you lose types and you enhance that with higher reproduction rates. So due to this, in each expansion direction, you lose one type, and the correlations between the types lost in like closed directions are going to give rise to sectors. So in particular, that means that, to like really understand this phenomenon of operation of sectors, we need a stochastic model. We can't just have a deterministic expansion. And while we understand things fairly well from a qualitative viewpoint, when it comes to actual like quantitative results on, like, for instance, the properties of the sectors, then there's basically nothing at the moment. So we don't know how many sectors we expect. We don't know their properties, their size. On what time scale do we see them? Yeah, for really like theoretical mathematical results, that's almost nothing. Part of the reason why is because, since until SLFVs, you almost had no way to study genetic diversity for populations living in a spatial continuum. So now you have SLFVs, but they only allow to study expansions of, for instance, advantageous types in an already established population. So it's not the same kind of expansions. You don't really have population genetics processes like specifically for expansions in the spatial continuum. And if you go like out of population genetics and go check what people did, for instance, in first passage percolation, then you can find a lot of stochastic growth models. And you also have some results about like coexistence of types, for instance, during like population expansions. But you don't have all the tools that you have in population genetics, like related to like duality relations and reconstruction of genealogies in particular. So it's like harder to tackle the questions regarding sectors. So what we would like is a model like combining the best of both worlds, like all the results we have in first passage percolation to study growth dynamics, and all the tools we have related to SLFVs to study genetic diversity. And an attempt to do that is the infinite parent SLFV that I will introduce in this talk. So just to give like few words on the type space and everything before giving you the model dynamics. So compared to what Amandine presented in like the previous talk, we need to like enrich the type space to add one type for empty areas, which we will just call this type empty. And moreover, like we'll need to consider a slightly simplified setting whenever we consider a location, it's either completely empty, either completely full, and with individuals of the same type. So at a specific location, you don't have several different types. That's kind of a technical assumption that we need to construct the process. And since each area is either completely occupied, either completely empty, and we can only have one type, it doesn't really make sense to consider like impact parameters anymore. So we have a Poisson point process, which is slightly simpler than earlier because we still have like the time coordinates, the coordinates of the affected area, but we no longer have a component for the impact factor. And as earlier, we would like the distribution of the radiuses of reproduction events to satisfy this condition, which ensure that if you take a compact area, like a bounded area, it is affected by reproduction events at a finite rate, which is like a reasonable assumption for the model to be well defined. So now to give you like a kind of informal definition of the process, we consider that initially we have some empty areas, which are like in white, and some areas full with like green individuals, like blue individuals, etc. And we are going to wait until our Poisson point process gives us a first reproduction event. So let's say that the first one is this one. Then what we do is that we check whether this event intersects an occupied area. Here, it's the case. And like all the individuals in the intersected affected area have the same type. So we'll just choose one of them. And this individual will reproduce and fill the area with its descendants. So we obtain something like this. Then we wait for the next reproduction event. And for instance, if it falls in a completely empty area, then there is nobody around, so no reproduction. And there is no visible effect. And the most interesting case is probably when you have like a reproduction event intersecting like occupied areas with individuals with different types, because then we need a rule to choose who is going to reproduce. And what we do is that we say that we sample the branch reproducing uniformly at random in the intersected occupied area. So either here or here, and with a probability that depends on like the respective volumes of the areas here and here. So let's say, for instance, that we sample the parent here, so it just reproduces and fills the area with its descendants. Okay, so of course, like this construction, as I said earlier, it's informal. It works on like a computer screen because the computer screen is bounded. But in practice, we want populations living in RD, so that means it's unbounded. That means in particular, we have an infinite number of reproduction events at each instance, and that this Poisson point process construction cannot be used directly. So we need a way to formalize it. So Amandine like mentioned 2 possible ways to do it in her talk. And that's like the first thing we would like to check, because if we want to be able to use like SLFV specific tools, we would like to have a process looking like the other ones. So, I mean, the easiest way generally is to try to characterize the process as the unique solution to a martingale problem. And to do that, we need the martingale problem to be well posed. So that means we need a dual process to like guarantee uniqueness. Then there is an alternative way via particle construction. And this one is also going to need actually the existence of a dual process. So we can't really escape that. So if we can define a dual process, then we have 2 ways of defining our infinite parenthesis SLFV rigorous. So we can have a look at how to do that. So to construct a dual process, the idea is to start from one area like that we take and ask ourselves whether this area is completely empty or at least partially occupied. And to do that, we can decide to just go backwards in time until we reach the last reproduction event which affected this area. So let's say that it was this reproduction event. Then we have 2 possibilities. So if the affected area was partly non-empty, then we had reproduction. And so this area here was completely filled after reproduction. So we know that at the present time, the area in gray was at least partly occupied. And the other case is that the affected area was completely empty. So then we just need to check the remainder. So in both cases, we can see that we have an equivalence between like the area we started from being partially occupied after reproduction and the bigger area being partially occupied before reproduction. So we are just going to add these reproduction events to our dual process. And we will continue going backwards in time until we have like new reproduction events, like intersecting the process. And we just add them until we reach the initial condition and conclude. And so we can see that over time, like we add more and more reproduction events to our process and we never remove them. So the jump rate is just going to increase and increase. And we need to be able to control it so that we don't have like kind of explosion in finite time for the process to be well defined. And once we have that, if we manage to control the jump rate, then we have our dual process. So we have the definition and the unique solution to a martingale problem or via particle construction. However, it turns out that it doesn't work that well. If the radius of reproduction events is bounded, then we can show that the dual process is well defined. Otherwise, it's still an open question. We do have a technical condition, which is like more general than the radius being bounded, but less general than the condition we would want, for which we can guarantee that the dual process is well defined. But we have a gap like between this technical condition and the one we would want, for which we can't guarantee that the dual process is well defined. And so we can't define the infinite parenthesis SLFV, which is a bit of an issue because like the construction, I mean, the condition of having an area affected by reproduction events at a finite rate seems fairly reasonable. So we would like it to be satisfied for the infinite parenthesis SLFV. And to do that, we are going to need an alternative construction of the process. And the one I will present in this talk is just based as defining the process as the limit of other processes which are well defined under our condition and which are called k-parent LFVs. So that's what we are going to see. So just before going with the details of the alternative construction, I would just like to spend few slides to give a link, a quick link with continuous first passage percolation, which are the other big family of models for stochastic growth. Because the model can seem fairly close to what is already done in first passage percolation. And in particular, when you consider a bounded initial condition, which is a fairly restrictive setting compared to what you can do with the infinite parenthesis LFVs. But when you consider bounded initial conditions, then the model is very close to one introduced by Maria Deschwen in continuous first passage percolation. However, you do have two differences which are at first only slight, but have big effects on the model dynamics. The first one is that in the infinite parenthesis LFV, you have reproduction when your reproduction events intersect an occupied area. In the model of Deschwen, you also need the center to be inside the occupied area. Having just this is not sufficient to have reproduction. And that implies that in the infinite parenthesis LFV, you have much more reproduction at the front edge, at least twice as much, generally even more. So you expect different growth dynamics. And on top of that, in the model introduced by Deschwen, once an area is populated, it kind of stays frozen in the same state. You only have reproduction descendants going into empty areas. While in the infinite parenthesis LFV, you kind of always have reproduction, even in completely occupied areas. So if you have existing sectors, they can be erased on a diffusive time scale in the infinite parenthesis LFV. But not in the model of Deschwen. So you have these two very small differences, but which can potentially yield very different growth properties and genetic diversity patterns. And on top of that, the infinite parenthesis LFV is more easily reversible, which makes it easier to have genealogies and things like that, and use population genetics tools. So the models may look similar, but actually not that much. So anyway, that was the end of the side remark on first passage percolation. And we now move on to actually giving a formal construction of the infinite parenthesis LFV. And to do that, we are going to introduce another process, which is the k-parenthesis LFV. So the informal dynamics is very similar. You still have these empty areas and these reproduction events. But when you have reproduction, instead of checking whether the complete affected area is partially occupied, we are just going to sample locations uniformly at random in the affected area. So we first sample a first one. If it's an empty location, we discard it. If it's an occupied location, then we say, okay, we found our parent, and this individual reproduces just as in infinite parenthesis LFV. So you fill the entire area with descendants. So if the location was empty, we are going to sample another one and repeat the process again and again until either we find finally an actual parent and an actual occupied area, or if we just give up at some point and say, okay, there is nobody, and we just kill all the individuals in the area. So in the k-parenthesis LFV, we give up after sampling k locations, hence the name. And you can also actually interpret this process as kind of an SLFV with strong selection against empty area, except you would have the same model if you wanted to like study strong selection and the spread of like strongly advantageous type. But instead of having occupied and empty areas, you will have like areas occupied with individuals with the strongly advantageous type and others. So anyway, we have our process, and it turns out that we can also define the dual process for the K parentheses LFV. We are going to have like potential ancestors growing in time. But contrary to the infinite parentheses LFV, the dual process is in fact well defined under the condition introduced earlier. And in fact, you can bound from above the jump rate by the one of Yule process with K descendants. So it's easy to show that it's well defined. And so usually this process is going to be defined like as the unique solution to a martingale problem. But that's not the best setting to like study a limit when K grows to plus infinity. So we are going to like introduce an alternative construction which will allow us to couple K parentheses LFVs and see what happens. So to do that, to make notations readable, I will make the simplifying assumption that we just forget about genetic diversity and just focus on the growth of the occupied area. But you can add genetic diversity into the mix after. And for our alternative construction, we are going to extend our Poisson point process to just add more information inside the Poisson point process about reproduction. Because at the moment, we only have the time and the affected area, and then we sample after the locations chosen for potential parents. And what we do here is that we extend the Poisson point process in order to like choose in advance which locations are going to be sampled. And in order to be able to couple different, like for instance, the 2 parentheses LFV with the 3 parentheses LFV and the K parentheses LFV, we're just going to take an infinite sequence of potential parents and choose after which one we take. Like for instance, for the 2 parentheses LFV, we will just take the first few potential parents given by the construction. If we, for K parentheses LFV, we'll take the first K parents. Okay. And the last thing we will need is regarding the initial condition. So rather than have something measure valued, we will need to switch to something density valued to ensure the coupling is possible, which can be interpreted as the indicator function of the area initially occupied. Okay. So to perform our construction with the extended Poisson point process, we can notice that once we have this Poisson point process, then the evolution is completely deterministic. All the stochasticity in reproduction is encoded inside the Poisson point process. So we have all the information we need in it. And in particular, we can use it to reconstruct the genealogy of an individual at a specific location at a given time, or at least by genealogy, I mean genealogy of potential ancestors. And that's going to give us what I will refer to as K-ancestral lineages, which are the sets of potential ancestors of individuals at a specific location at a given time. So here, since we only care about whether an area is occupied or not, we only need a set. But if we were adding genetic diversity, we will need to have an ordering also in our sets. But that's not the case at the moment. So to construct like this K ancestral lineage, we do like exactly what you expect. So it's like a lot of notation to describe something fairly easy. So you start from your location Z at time t, and you go backwards in time in your extended Poisson point process to try to find the last reproduction event which affected this location. Then you are going to look into the sequence of potential ancestors to take the first K ones, which are going to be the ones chosen in the K parentheses LFV. And you say that one of them can be seen as the actual parent. So we just add them all and remove the descendant. And we repeat that again and again. We go backwards in time. We try to find reproduction events which affected at least one of the potential ancestors. And then we remove descendants and add potential ancestors again and again. So now, regarding whether this process is well defined, if we kind of do things unconditionally, that makes sense, and just forget about the Poisson point process, we already saw earlier that it works, and we can just bound the jump rate from above by the 1/U process with k descendants. Contrary to like the infinite parental levy, you no longer have the issue of like very big reproduction event just making the jump rate completely explode. Here it's quite the opposite, in fact, because like very big reproduction events are likely to make a lot of lineages coalesce and decrease significantly the jump rate. So yeah, everything works well. However, when we kind of add the Poisson point process into the mix, things get a bit different, because you can have an issue if your Poisson point process is in the realization of it, at least, is in such a way that you have two reproduction events affecting the process simultaneously at some time before you reach the initial condition. So if you just consider one location at one specific time, then you know that it will almost surely never occur. But we are repeating this construction for all locations in RD and at each instance between, after 0, so times 0, so after the initial condition. So we may have some issues occurring that we need to take care of. And we will say that the realization of a Poisson point process is good if there is no location which is affected simultaneously by two reproduction events at some time. And it turns out that we can show that the Poisson point process is almost surely good. And to do it, we can just like cut RD into rectangles and say that for things to go wrong, we need like one of these bounded rectangles to be affected by two reproduction events simultaneously at some point during a bounded interval. So it just gives us an upper bound, and we end up with a countable sum of events of probability 0. So yeah, so it almost surely never occurs. We can like almost surely like define our ancestral lineages for each location at each time, due to the correlations induced by the fact that we work with the same Poisson point process at all times. So now that we have that, all this framework of K ancestral lineages, we can finally introduce our K parental LFV in an alternative way. And we will say that like a location is occupied if at least one of the potential ancestors at time 0 is in an occupied area. And if we want to add genetic diversity, then we will have an ordering of the ancestors, which will allow us to decide the type. So we end up with two ways of defining our process: as a density valued process or as a measure valued process. And our measure valued process is also the unique solution to the martingale problem, which is usually used to define the K parental LFV. So this alternative construction gives the same process as if we had done as usual, which is good. But so can seem like to not do much. However, contrary to the martingale problem construction, it is much easier to couple together different K parental LFVs with, like, for instance, a different number of potential parents, because they are all constructed using the same Poisson point process. And in particular, like, since we always choose the same first potential parents, that means that each potential ancestor of an individual in the K parental LFV is also going to be a potential ancestor in the k+1, k+2, etc. Parenthesis LFV. So that implies, just by construction, that if an area is occupied in the k parenthesis LFV, it's also going to be occupied in the k+1, k+2, etc. SLFV. So for each location and at each time, we have a sequence which depends on k, which is non-decreasing and that values in like the set 0, 1. So it's going to converge. And we define our infinite parent SLFV as the limit, like the function whose value at each location and each time is given by this limit. So that's our construction. And again, we can have two viewpoints on our process, as a density valued process or as a measure valued process, which we need later. So now that we have that, we can see that there are few results on the dynamics of the infinite parenthesis LFV, which can be directly recovered from the construction. So first, we can see that the value taken by the infinite parenthesis LFV of our bounded set is going to be updated at a finite time, at finite rate, sorry, which is something good, but which is not always that obvious in the martingale problem construction, for instance. Here it's just a direct consequence. Moreover, we can show that our density is measurable. So we can also interpret our process as a set valued process rather than a density valued or measure valued process. However, there is something which is a bit funny, is that the construction does not give us directly that that basically once a location is occupied, it stays occupied. Like the density at location Z is, I mean, the fact that it's increasing is not a consequence of the construction. So if we only have that, I mean, we need much more work to actually have this result, which we will expect from the informal construction. So this is something we considered with Amandine in like a preprint, which is an archive. So it turns out that when the radius is bounded, then we can show that, I mean, for a given Z, it's almost, I mean, the sequence is almost surely non-increasing. So once you are occupied, you almost surely stay occupied. And we are working with like Matt Roberts, so my postdoc supervisor, on trying to extend it to like having almost surely for all Z. But anyway, so this can seem like nitpicking at first, but it means that the interpretation of the process as a growing random set rather than just a random set is far from being straightforward compared to what the informal construction gives us. So then, the coupling can also be used to like obtain like several kind of convergence results. We have a kind of pointwise convergence of the k parenthesis LFV towards the infinite parenthesis LFV, which is just like the definition of the process. But we can also actually go further than that. And at each time t, we can show that the measure describing the k parenthesis LFV at time t converges almost surely to like the limiting measure. And we actually have something even stronger just using this coupling, which is like the convergence in distribution of the k parenthesis LFV towards the infinite parenthesis LFV. So I put here like the details of the proof, but there's like nothing original at all. It's just, I mean, very classical way to show convergence in distribution when you have a characterization as the unique solution to a martingale problem. So yeah, nothing new. However, that's like an example of how you can use like this alternative construction of the k parenthesis LFV and the coupling to obtain like a result regarding convergence in distributions. Since we have like impact parameter of 1 and we do not rescale time or space, this is not really kind of scaling regimes that you can have a look at with the techniques we saw in Amandine's talk. So you can see this coupling as like a new tool to study other limiting regimes in SLLV type processes. So then to kind of try to map everything back to the introduction. So the reason why we needed this alternative construction is because the usual construction of the SLFVs didn't work in the more general setting. However, we did have like restricted settings in which the usual construction works. So we would like to check that our process corresponds to what we would expect in that cases. And it turns out that everything works well if you can define, I mean, if the martingale problem we wanted to introduce earlier is well posed, then our infinite parenthesis LFV is going to be a solution to the martingale problem. And so like, in fact, the unique solution to this martingale problem. And moreover, if the area initially occupied is bounded, then you can just define your process as a set-valued self-dual process, which also corresponds to this process we saw as the limit of K-parenthesis LFVs. And is like closer to what you have in continuous first passage percolation. And in particular, is actually a growing random set, contrary to the infinite parenthesis LFV in the construction as limit of K parenthesis LFVs. Okay, so then to conclude, like, what next? Because like I kind of sold this model at the beginning as something to study genetic diversity in expanding populations. And we spent the whole talk looking at like very technical results regarding the process. So you may think that, yeah, but didn't really keep my promise. But actually, like, that's kind of the perspectives regarding this work, how to use this process to like study genetic diversity in expanding populations. And to do that, the first step is to just understand the growth dynamics. Because we saw earlier that the process looks like others you have in first passage percolation, but it's not close enough to be directly able to use the results we already have. So, and in particular, you also need to find a way to transform your measure valued process into something on which you can apply like percolation techniques. So that's something like in the article with Amandine. And moreover, something which is more of interest to study genetic diversity in the at the front is actually genetic. I mean, the growth of the area with like maximal density at a mesoscopic scale. So rather than just the front edge where you have like densities at a mesoscopic scale, which are going to be non-zero, but not one either, some kind of intermediate, we like to be slightly behind where we have maximal densities. Because once we are there, then we can be in a setting which is like much closer to just like populations already well established in the spatial continuum and forget about the expansion part, which makes things easier. So that's something I'm working on at the moment with my postdoc supervisor. And we are also looking like at other things regarding the actual speed of growth, the effect of stochasticity. So it's just more simulation-based work, which is fun. And once we have that and understand the growth properties of the occupied area, the long-term goal is to study the aberration and persistence of sectors by looking at like genealogies of individuals conditioned on being in occupied areas, and preferably in areas of maximal densities, but close to the front, which are the one in which we want to be, to observe sectors, but still having good densities. So this won't really involve the coupling, but it can also be, this coupling can also be used in other settings, hopefully to like explore other limiting regimes regarding spatial non-Fleming process. And that's it for me. So thank you for your attention.
Thank you very much, Apolline. Are there questions?
So I have one question. You mentioned connections to first passage percolation.
Yeah.
First passage percolation and a lot of these growth models have connections to the KPZ equation.
Yes, that's a very good question. That's something we looked at numerically.
And so do you believe that your model is in this KPZ universality class?
So we kind of explored like the front fluctuations, spatial fluctuations at the front of the front position at specific times. And so far the results we have seem to fit with being in KPZ universality class. So of course it's not sufficient to demonstrate it, but it does not contradict it so far. We will expect also the model to be in the KPZ universality class because it looks a lot like the Eden-Groth model, which is also conjectured to be in the KPZ universality class. So yeah, we looked at it numerically and it doesn't contradict being in the class.
Okay, thanks.
Are there any other questions? Andre?
Thank you. I think that in some other articles, spatial Fleming-Wyatt process, lambda Fleming-Wyatt process is connected to branching Brownian motions. Could this be done also here, say in the scaling limit or anything similar?
So here we wouldn't be able to get Brownian motions first. For two reasons. The first one is that we are not looking at a kind of scale, temporal and spatial scale at which you expect to see Brownian motions in the limit, because to have them, we will need to do a diffusive rescaling. But we are looking at an expansion which is linear in time. So that's the result we have with Amandine on arXiv. So if we do a diffusive rescaling to recover Brownian motions, then we lose the expansion because it will occur like immediately in the limiting regime. So that's the first reason. And the second one is that, so actually, if I come back to the definition of the key parent SLFV, so like the regime for the SLFV in which you get like kind of branching Brownian motions is when you have two types of reproduction events: neutral reproduction events in which you only choose one parent, and selective reproduction events in which you should, for instance, k potential parents. And to see branching, you need like the rate of selective reproduction events to be much smaller than the rate of neutral reproduction events. So that's in the limit, you have your lineages moving like Brownian motions, and then you have like some finite time between two consecutive selective reproduction events in the limit. But here, that's like the same setting, except we no longer have neutral reproduction events and we only have selective reproduction events. So we have no hope of seeing like branching Brownian motions.
This holds for K and for infinity parent.
Infinity parent, it's even worse. Yeah, it's like the complete opposite regime.
Okay, thank you. Thanks.
I have another question. I was wondering if somebody tried some kind of opposite model where, for instance, well, it's not relevant for your bacterial example, but if you think about a species with a given range, then the edge of the species might be less suited for it. So in a way, it would not reproduce as well. Is it just that at equilibrium there's no more expansion and it doesn't collapse. So I don't know what you predict in this case.
So I think like for this, the first challenge will be like trying to like implement that in terms of spatial lambda Fleming processes in a way that kind of makes sense for the process, and that I guess that will be something worth looking at, but I'm not really able to do like the modeling part on the spot. Sorry.
Thanks.
Yeah.
Are there more questions? So thank you very much, Apolline. Thank you very much, Amandine. This was a very nice meeting.
Thanks again for the invitation, and for everyone for being patient and staying until the end.
And we will meet again in 2 weeks for the talk of Milton Jara.
And Ottavio Menezes.
Ottavio Menezes, yeah, sorry. Okay, so
Bye.
Bye-bye.